Okay, so what we're going to do is we are going to look at a sheep brain in terms of dissection. And I actually do have a model of the human brain as well that we're going to use kind of to compare some of the structures. So I want to talk about the human brain first so we can kind of look at bigger structures and then look at the smaller on the sheep brain. So the major portion of the brain is the cerebrum. And then this guy back here, this is the posterior aspect, is the cerebellum. So when we flip it over, this is what a dissected brain would look like. And that's what we're going to see with the sheep brain. So once again, all of this is the cerebrum. This here is the cerebellum. The other things that we're going to see is we're going to see the diencephalon, which is here, our thalamus, which is right at the top of the brainstem. We have our epithalamus above that. We have our hypothalamus, and then we have our pituitary gland. The other thing that we're going to see is we're going to see this structure here. This is the corpus callosum. That's going to connect the two sides of the cerebrum for communication. And then when we get into the brainstem, we have the midbrain here. We have the cerebral aqueduct, which is basically this structure here. We have what's known as the corpora quadrigemina. There's 4 protrusions. There's 2 on either side, so we see 2 of them here. We see the pineal gland, which is going to be here. And then we get into the pons, the medulla oblongata, and then that's going to go into the spinal cord itself. Another thing we're going to see on the sheep brain is we're going to see this structure right here. This is called the olfactory bulb. Olfactory bulb is actually part of the olfactory nerve. It goes into the olfactory nerve, and that is for the sense of smell. And we're going to notice a big difference in the size of the olfactory bulbs between sheep and humans. One of the things that you're going to notice is that the structures might be slightly larger or slightly smaller on the sheep brain, and depending on how big the structures are depends on what we're using it for. So first thing I want to mention here is when we look at the cerebellum down here, you'll notice that the cerebellum makes up about 1/5 of the overall size of the brain. On the sheep, it's a lot bigger. And so the sheep are basically going to use their cerebellum for more. So let's get into the actual brain itself. So when we're looking at the brain, I basically have 4 different parts. I have 2 full brains, I have a dissected brain, and then I have a section of the brain. So I want to start with this brain here, and you'll notice it looks a little different because it's all got this white connective tissue on it. So if you remember the meninges are there to help protect the brain. The most superficial meninge is going to be this dura mater, and it's actually really tough, and you'll notice it's connected to the brain. It kind of goes and folds into the brain. It separates structures, and then underneath that, which I don't really see very well, you can kind of see parts of it here. So you can kind of see that it kind of looks like a spider web. That is the arachnoid matter, and then on the surface of the brain itself, there's a bunch of connective tissue, and I'll actually break it on one of these other brains here in a second, but that is the pia mater. The pia mater is going to be attached to both the brain and the spinal cord, and all 3 meninges, both the brain and the spinal cord, actually have. So when we take a look and we've taken off the dura mater, this is what the brain's going to look. This is basically top, bottom, anterior, and posterior. So when we're looking at this, first thing you're going to notice is this very large cerebrum. This back here is the cerebellum, and then we're going to start getting kind of into the spinal cord down here. So when I look at the brain, you'll notice there's all this black stuff. That's actually the blood vessels. And you'll also notice that the brain itself is not smooth. It kind of has these indentations, which are called the sulci, and these projections upward, which are called the gyri. The other thing that you're going to notice is we have some natural separations, okay? These natural separations are called fissures. So whenever I'm looking at this, there's a couple of things that I'm going to be looking for. So you remember me talking about the olfactory bulb on the human brain. This is actually it on the sheep brain. So you notice it's a lot larger, and the reason it's a lot larger is sheep are going to use their sense of smell a lot more than we are because they have to find food, but they also have to avoid predators, okay? The other things that we're going to see here when we're looking at this side of the brain is we are going to see, and it's more visibly seen on this one here, this is the optic chiasma. So the optic nerve is actually going to cross over to go to, the opposite side of the brain. So you can see this right here. This area right down here is actually where those two cross. That's the optic chiasma. It's been kind of broken down here, but you can see remnants of it right here and right here. So that's the optic chiasma. So underneath that, we're going to see this section right here. These are the mammillary bodies. And right in the center, you're going to see this area. It's called the infundibulum. That's actually where the pituitary gland used to be. So this thing right here is the pituitary gland. So right underneath the optic chiasma, we're going to have this pituitary gland. And that's more part of the endocrine system. So when we talk about the endocrine system, we'll get there. We are then going to kind of move down. So from our mammillary bodies, we're going to go down here. This is our pons. And then our pons is going to give rise to the medulla oblongata and then into the spinal cord itself. So I talked about pia mater, right? So you can see this has been naturally separated. If I continue kind of pulling on it, that's one of those fissures. It's called the central fissure. It separates the left and the right side of the brain. So as I'm pulling that, you'll notice that there's tissue in between that's kind of holding the brain together. That area there is called the corpus callosum. That's what's actually connecting both sides of the brain for communication. Remember that the left side of the brain controls the right side of the body, right side of the brain controls the left side of the body, so there's got to be communication. So now we can actually look at the dissected brain itself. So first thing that I'm going to do is I'm going to kind of pull on this, and you'll notice that there's this cavity here. That is one of the ventricles of the brain, and normally those ventricles contain cerebrospinal fluid, and they're going to be lined with ependymal cells. Those ependymal cells are actually circulating that cerebrospinal fluid. Right underneath that, right here, is going to be the thalamus. Remember, the thalamus is a relay station. It's sending information to the appropriate part of the cortex, the cerebral cortex, for interpretation. And it's right at the top of the brainstem, because as information comes up the spinal cord into the brainstem, it's going to hit the thalamus, and then it's going to send that information to the appropriate part of the brain for processing. Down here would be where our hypothalamus is, and then our pituitary gland, the remnants of it are right here. So moving down into the midbrain, I want you to notice that if I kind of pull the cerebellum apart from the cerebrum, as we're looking into this area, you're going to notice basically these four protrusions, 1, 2, 3, 4. That's the corpora quadrigemina, and we can actually see two pieces of it here. Corpora quadrigemina is used for auditory and visual reflexes. As we move down, this little area here, this bump here, is the pons. It's quite a bit smaller in us, and quite a bit smaller in sheep than it is in humans. From there, we're going to go into the medulla oblongata and then into the spinal cord itself. So that's the spinal cord. The other thing we're going to see is we're going to see the cerebellum. So remember how we compared the size of the cerebellum in the human brain. So you'll notice that the cerebellum in the sheep brain is about 1/3 of the size of the entire brain, which means they use their cerebellum a lot. Their cerebellum is specifically for balance, and they're four-legged animals, so they're going to need a lot better balance than we would. The other thing I want you to notice is you notice the white matter and the gray matter of the brain. So the white matter in the brain is actually deep, and the gray matter is superficial. Our white matter is white because it's made out of myelinated axons. Those axons are sending signals. The gray matter is mainly made out of cell bodies. There are some unmyelinated fibers as well, but it's mainly cell bodies, and that's where the processing is going to occur. The other thing that I have here is a slice of the brain. So what I did was I took one of these brains and sliced it this way. And so that way you can see that the gray matter, once again, is superficial in the brain, while the white matter is deep. Once again, white matter made out of axons sending information. Gray matter is going to be superficial, and it is basically processing information.